All right, folks, so let's talk about the functions and special features of the muscles. So muscles represent around half of the body mass. The main function is to convert the chemical energy of ATP molecules into movement. It can be the movement of bones and can be movement of fluids, for example, cardiac and smooth muscles. Exactly that. Obviously, postural contributions, all of that. Second really important function of the muscles is to produce heat, which is achieved by skeletal muscles the most. And a bunch of other accessory functions such as regulation of opening and closing of the valves, the eye accommodation, the size of the pupil, and so on. Muscles have three features that distinguish them from other types of tissues. First of all, muscles are excitable, which means they can be stimulated and will respond to that stimulus, usually through contraction. They are contractile, that's the only tissue that can forcibly shorten upon the stimulation, and they are elastic and extensible, which means muscles can be stretched. As you may remember, there are three muscle types: skeletal, cardiac, and smooth. So what are the differences between the three? Well, first of all, the only muscle type that can be contracted voluntary is the skeletal muscle. Cells differ anatomically because skeletal muscle cells have many nuclei. Cardiac muscle cells usually have one, sometimes two, and smooth muscle cells have only one. Unlike smooth muscles, skeletal and cardiac are striated, which means they have this visible stripes that, as we will learn in some time, are defined by the basic contractile units of these muscle types. The only branching type is the cardiac muscle, which makes sense because heart has to contract not in the linear but in three-dimensional fashion. And then they differ in terms of the stimulus that can trigger the contraction. So skeletal muscles can only be activated by the nervous stimulus, which makes sense since nervous stimulation is the only one that we have voluntary control over. Unlike skeletal muscles, smooth and cardiac can be stimulated by a variety of agents, from nervous and hormonal to chemical and mechanical. These three muscle types also differ in how fast they contract. Skeletal muscles are the fastest. Cardiac muscle contracts slower, and the slowest of them is the smooth muscle. In this specific unit, we're going to focus on skeletal muscle and a little bit on the smooth. So first, let's talk about skeletal muscles. Overwhelming majority of them are attached to a bone or two bones or a bone and skin. When we use the term skeletal muscle as an organ, we divide them anatomically. So basically your biceps brachii is an organ. It's one of many skeletal muscles. Usually every skeletal muscle is innervated by a single nerve. Blood is supplied through a single artery and drained by one or more veins. Skeletal muscles appear red, as you all know, and partially it is because of a lot of blood vessels. In other words, dense vascularization. The reason for this: skeletal muscles, when active, require a lot of nutrients, and they also need the removal of a lot of waste. As we will learn again, but basically your skeletal muscles consist of many cells, linear cells, that we call fibers. Every muscle fiber has a dedicated nerve ending that controls it. All muscle fibers that are controlled by a single neuron are called a motor unit. Here on this image, you can see that this axon controls multiple muscle fibers. For instance, this, and perhaps this. When we are talking about the attachments of the muscles, we use the term insertion and origin. Origin refers to a body part that remains relatively not moving during that muscle contraction, and insertion is the bone or skin that moves. I can give you a couple of examples. For instance, biceps brachii, the muscle of the flexor of your elbow, well, flexes your elbow. When that happens, your shoulder, more specifically scapula, remains calm, stays in place, stays in place. So the origin is scapula. The bone that moves is radius, so the insertion is radius. This is an example of an insertion with two bones. You have a muscle called frontalis. Sometimes it's called the frontal belly of the occipitofrontalis muscle. That muscle raises your eyebrow. So when you are looking surprised, your skin on your forehead moves up because that muscle contracts and pulls the skin towards your scalp. Now, muscles attach to the bones via periosteum. Periosteum. Remember, each bone has a layer of connective tissue called periosteum. And muscle also, every muscle as an organ is surrounded by similar connective tissue called epimysium. So when epimysium directly fuses to periosteum, we call it a direct attachment. When muscle is connected to the bone via a tendon or an aponeurosis, that's called indirect. So now let's go over the anatomy of the muscle. So first, muscle, an organ such as biceps brachii. The entire muscle is, let's use the word encapsulated, in the connective tissue called epimysium. Remember, epi means above. Epimysium is the dense irregular connective tissue, and it provides mechanical resistance to the muscle. Every muscle as an organ consists of thousands of muscle fibers, individual cells, and these cells are organized in bunches. Every bunch of cells is called a fascicle. I don't know if you wonder, but if you wonder whether the word fascist has the same origin, the answer is yes. Fascicle in Latin means a bunch. I'm not going to go into etymology of the word fascist, but they all come from Latin term for a bunch. Every fascicle is surrounded by the fibrous connective tissue called perimysium. Still dense connective tissue. When we will look inside the fascicle, we're going to find many muscle fibers, individual cells. When we use the word muscle fiber, well, not the word, but expression, muscle fiber, it's the same as the skeletal muscle cell. Each cell is wrapped very gently into connective tissue called endomysium. It's a fine, loose areolar connective tissue. I want to warn you against one common mistake / confusion. As you know, every cell, any cell, its boundary is cell membrane. Every muscle cell has a membrane. Outside of that membrane is endomysium. So, when we start looking deeper into a muscle cell, what we're going to find is that it contains a lot of cylindrical myofibrils that are penultimate contractile structures. Basically, the elements that form every myofibril, so-called sarcomeres, are elementary contractile units of a muscle. Skeletal muscle cell, or muscle fiber, as you can imagine, structurally is adapted to perform the most important function, contracting and moving things. There is some special terminology. The reason for having that special terminology completely evades me, because I think the common terms are just as good, but I always mention that anatomists definitely deserve their special place in hell. Okay? So, muscle fiber, long and cylindrical, pretty thick, diameter-wise, up to 100 micrometers. Technically speaking, you can take a piece of muscle tissue and using a microscope, and you don't even need the most powerful microscope, you can tease it into individual fibers. Because 100 micrometers is actually thicker than a human hair. It can be very long, up to 30 centimeters. Imagine human sartorius muscle, which is located in the anterior aspect of your upper leg. It's the longest muscle in the human body. It's about 30 centimeters. Since skeletal muscle cells are generated when multiple myoblasts, the precursors, fuse, skeletal muscle cells have multiple nuclei which are pushed to the periphery of the cell, giving the space for myofibrils. The term that is used for plasma membrane of skeletal muscle is sarcolemma, The term, basically everything with myo, mys, or sarco refers to a muscle. Sarcoplasm, cytoplasm, sarcoplasmic reticulum, endoplasmic reticulum. A little note, sort of keep it in mind. Endoplasmic reticulum is the place for calcium storage. It will become important when we will talk about the contraction process. Glycosomes are not membrane-bound organelles. Those are special bodies that store glycogen, which can serve as the fuel for the muscle contraction. And finally, muscles are full of the protein called myoglobin. It's a relative of hemoglobin found in your blood, and its function is to store oxygen, just like hemoglobin does. But hemoglobin is transport, myoglobin is storage, so there are some differences. As we already discussed, each myofibril is shaped like a rod and consists of multiple basic contractile units called sarcomeres. I usually suggest for students to think about a myofibril as a train that consists of multiple cars, and each car is a sarcomere. Well, let's take a look at the composition of a sarcomere. So an individual sarcomere is bound by two Z-discs. Sarcomere itself consists of several different types of filaments, and each of these filaments is composed of its own specific type of a protein. In the center of the filament are several filaments that form what's called an M-line. It is an anchoring point for so-called thick filaments. The thick, red thick filaments that are attached to the M-line consist of a protein called myosin, while the blue, thin filaments that consist of a protein actin are attached to Z-discs. Thick filaments are indirectly attached to Z-discs as well via elastic filaments composed of a protein called titin. When you look closely at the structure of a sarcomere, you may notice that there are areas where thick and thin filaments overlap. So effectively here, these filaments overlap. In the meantime, elastic filaments are very thin, even thinner than thin filaments. So this area is where there is no overlap. Overlapping filaments appear under the microscope as a darker stripe, so-called A band. That's a bit too much, I think. While the areas where thin filament doesn't overlap with thick one, called I band. This is the explanation of the striated appearance of a skeletal muscle. The A band is dark, while the I band is light. In addition to these proteins, there are a couple more. I will not ask about those in the exam. I just want you to have sort of a connection. So nebulin is the protein that regulates the assembly of thin filaments, and the dystrophin, very important protein in the context of human diseases, we will see later. Dystrophin connects thin filaments, and sarcomeres stabilizes the structural integrity of a muscle fiber. Any questions so far? I'm not going too fast, am I? Let me know if I'm going too fast. Too fast? Okay. So now let's compare, or I'd rather say just look at the structure of thick versus thin filaments. So thick filaments consist of the molecules of protein called myosin. Each myosin molecules contain two heavy chains that form the tail and four light chains that form heads. As you can see, tails themselves form a body of a filament. This rod-shaped structure basically tails. Now the heads are sticking out, as you can see on this picture. Turns out the heads have two main functions. One of them is to bind ATP and to break it down, so-called ATPase activity, to release the energy required for the contraction. Another function is to bind to actin filaments, to thin filaments, forming so-called cross bridges. Now, unlike thick filament, which is more of a, fibrous, thin filaments mostly consist of the globules of protein called actin. When these globules attach to each other, they form these two interwoven spirals that you can see highlighted in blue. And on these spirals there are two more proteins. One of them is tropomyosin. If you look at this image very carefully, you may notice that tropomyosin blocks these little openings in these beads of actin. Those openings represent places on the actin protein that can bind to myosin heads. When tropomyosin blocks those places, the filaments, thin and thick, cannot interact with each other. Troponin is a protein that's attached to tropomyosin and controls its position. It can shift tropomyosin away so that sites for interaction open and myosin and actin can connect and form cross bridges and facilitate the contraction. So in heart attack, what you're referring to, when there is skeletal muscle and cardiac muscle structurally are similar in a way that cardiac muscle also consists of sarcomeres. It also has troponin, and when during myocardial infarction, cardiomyocytes inevitably die, troponin is being released into the bloodstream. Oh, it's not supposed to be in bloodstream at all. And the same story, I don't know if you are familiar with the TV show House MD. My favorite part of it is just to critique, because in many cases they stretch it a bit too far. But the main character, at some point, it's explained he had a muscle infarction. So the clot that blocked the blood flow to a skeletal muscle. In this case, his troponin levels would also skyrocket because it's similar troponin. So now we're going to return back to sarcoplasmic reticulum, that calcium storage place that I mentioned before. So sarcoplasmic reticulum is smooth ER, not responsible for protein synthesis, and its main function is to store and release calcium. Now, let's take a look at this image. If you want to better understand the mechanism of contraction, you have to turn on your imagination. I don't have a lot of analogies here, but I'll try my best. So, myofibrils and in this case, a myofibril will be this entire thing, okay? So imagine that these myofibrils are logs, like tree trunks, and you put them horizontally, like in the pyramid. Can you imagine that? Will there be spaces between the trunks? Yeah. So within those spaces is your sarcoplasmic reticulum, shown on this image, blue. Now, we have to stretch this analogy a little bit further. Imagine that you cut long pieces of relatively small, thin hose, like a tubing, okay? And you run it between the logs. So that, let's say you start pouring water, that's out of control. Analogy went out of control. But you start pouring water on one side of the pile, and it comes out on the other. So the idea is that tubing penetrates the entire pile of logs. So this tubing here is so-called T-tubal, shown here in white. You probably can't help but notice that every T-tubal is in direct physical contact with sarcoplasmic reticulum, more specifically with the structures called terminal cisterns. These are terminal cisterns of sarcoplasmic reticulum. So T-tubules structurally are extensions of the membrane, extensions of the sarcolemma, and they first of all increase surface area and provide the continuation of extracellular fluid into the lumen, okay, to connect the membrane with the sarcoplasmic reticulum. Why this is important? Okay, so, well, that assembly of two terminal cisterns of the sarcoplasmic reticulum and the T-tubule is called a terminal triad. Why this is important? So, on T-tubules, you're going to find dihydropyridine receptors. Of course, I will ask you 100% on the exam to draw the formula of dihydropyridine. Of course, I will not even ask the acronyms for the receptors. I want you to understand what's going on at the junction. On terminal cisterns, you're going to find other receptors called ryanodine receptors. In this picture, the receptors that I color with red is the HPRs, and the receptors that I color in blue are ryanodine receptors. These receptors physically contact with each other. Now, it's not going to be a secret for you folks that muscle contraction effectively is an electric stimulus in the beginning, right? You know that if you get electrocuted, your muscles are going to contract, right? So the function of the T-tubule and the terminal cistern at this junction is that the electrical signal that goes through the membrane of the skeletal muscle cell triggers the release of calcium from the sarcoplasmic reticulum into the cytoplasm, and that leads to the contraction. So, how does it contract, that skeletal muscle? Well, the model is called a sliding filament model. First of all, I want to mention that the distinctive feature of muscle contraction is that force is produced. Muscle fiber may actually not shorten. So let me give you an example. Imagine that you are trying to push the wall away. You won't be able to. There will be no movement. Is it a contraction? It's a contraction without muscle changing its length, because there's no movement. Okay? Now, if the tension that you manage to produce is greater than the load, then muscle will contract, obviously. But if the tension, the force that muscle produces is lesser, then there will be no shortening. So how does that work? Well, let's take a look at these two sarcomeres. So on the left, the sarcomere is relaxed. You can see that thick and thin filaments do not form cross bridges. There is no interaction. Okay? This is the width of the A band, and this is the width. I think it's not the great idea in terms of the color, so let's see if I can do better. Not really. So this is your A band, and that width here, that's the 1/2 of the I band. On the contracted sarcomere on the right, what you see is that there is physical contact between thick and thin filaments. I'm going to highlight it even more. This, 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 and this. During contraction, now hear me out, these cross bridges that are formed by the heads of myosin attached to the actin, these cross bridges effectively bend and pull thin filaments, and I'm going to use blue color to signify that it's thin filaments, pull them towards the M line. So filaments slide against each other. Okay? Now I'm going to try to employ one very cool technique. Bear with me, and if it doesn't work, not my fault. Okay? Let's see if I can do this. Yep. Wait, where's the ruler? Where is... I don't understand that. I'm going to do it one more time, and if it doesn't work, screw the ruler. Okay? I don't see no ruler. Okay, what I wanted to do, and thought it would be cool, is to measure the size of the filament. So I'm going to spoil that for you. If you would measure the size of each individual filament on the left and on the right images, filaments filament length stays the same. Filaments do not shorten themselves. They slide. So now when you look at both of these images together, you realize that when thin filaments are pulled towards the center of the sarcomere, Z discs come in closer. So these are Z discs, and you can clearly see that the distance between them, the discs, that distance is much shorter than that distance. Okay? However, the sarcomere becomes shorter, obviously. Okay? I bands also shorten. But A bands, that part, A bands stay the same because what determines the width of the A band? The length of a thick filament, and its length doesn't change. Okay, so there will be few questions about the sarcomere contraction. Now we're going to figure out how a nerve signal can cause contraction. And this happens during a process named excitation-contraction coupling. Coupling implies that one cannot happen without another and vice versa. So the result, if excitation is sufficient, there's going to be contraction. And contraction can't happen without excitation. If we look at it broader, every muscle fiber is innervated by at least one axonal branch. And that contact between the branch and the muscle fiber is called neuromuscular junction, or NMJ. When nervous system sends the signal, muscle fibers are activated on the neuromuscular junction. That results in the action potential on sarcolemma. Action potential eventually reaches T tubules, which you may remember are connected to sarcoplasmic reticulum, which results in the release of calcium from sarcoplasmic reticulum, and that calcium starts the contraction. Let's take a look at the neuromuscular junction. It is not any different from a synapse. So here you can see the axon of a motor neuron. When an action potential when it arrives to the axonal terminal, it opens calcium channels which results in the release of neurotransmitter from the synaptic vesicles into the synaptic cleft. The neurotransmitter that is released from those neurons is always acetylcholine. Now the membrane, the sarcolemma at the neuromuscular junction, is folded in so-called junctional folds. When you see folds, what's the purpose? Exactly, increased surface area, which means there are a lot of the receptors for acetylcholine. These receptors are basically ligand-gated sodium-potassium channels, and acetylcholine serves as a ligand. So what exactly happens? I'm not going to use the text. You can read it without me. Instead, I'm going to talk about the events at the neuromuscular junction. So first, as we mentioned, action potential arrives at the axonal terminal. That action potential opens voltage-gated channels that let calcium in, which results in the release of neurotransmitter acetylcholine into the synaptic cleft. This neurotransmitter diffuses across the cleft and binds. So once the neurotransmitter is bound to its receptor, remember it's a ligand-gated channel, this opens the channel allowing sodium to get into the cell, because remember, sodium concentration is higher on the outside, and simultaneously it lets potassium out, because potassium concentration is higher on the inside of the cell. Okay? Now, overall, there's more sodium comes in and potassium comes out. So overall, you have depolarization on the sarcolemma. As we discussed before, you have to terminate the signaling. You have to remove neurotransmitter, and as you may remember, acetylcholine is removed by enzymatic degradation. The enzyme called acetylcholine esterase degrades acetylcholine, and that results in the closing of the sodium-potassium channel. Because obviously if you don't terminate, then you're going to have constant contraction. So as we just mentioned, the diffusion of sodium into the cell results in the depolarization of sarcolemma and formation of so-called end plate potential. End plate potential is graded potential. You may remember graded potentials are different from action potentials, because graded potentials do have a value. If this end plate potential crosses the threshold of activation for voltage-gated sodium channels, shown here, okay, then remember my analogy about the domino. So once the threshold is reached, opening of that sodium channel will result in action potential, which will sweep through the sarcolemma. Just like with the neuron, opening of voltage-gated sodium channels results in depolarization, while the closing, more specifically, inactivation of sodium channels and opening of voltage-gated potassium channels results in repolarization. So we now arrived to the point when we explored how the stimulation at neuromuscular junction leads to the formation of action potential on the sarcolemma, which sweeps through it, and, well, eventually, get into the T-tubules. To summarize what we have in this case. So depolarization, I'm going to focus on the graph. So depolarization is due to the entry of sodium through voltage-gated channels. At the peak, sodium channels close. Not just close, they are inactivated. Remember, they can't be opened by anything for some time. Potassium channels open, and cell repolarizes because potassium diffuses out of the cells, and eventually these potassium channels do close. The restoration of the concentrations is done by, just like in neurons, by sodium-potassium pump. All this story is about the excitation. Right now we're at the step when muscle cell is excited, i.e., there is an action potential running across sarcolemma. Eventually, it will get into the T-tubule. And in the T-tubule, it will activate dihydropyrimidine receptors, the ones that back then I highlighted with red. The activation of DHPRs, these receptors, will stimulate ryanodine receptors, the ones that I highlighted with blue, and that will lead to the diffusion of calcium from sarcoplasmic reticulum into the sarcoplasm. Presence of calcium in the sarcoplasm is what triggers muscle contraction. So now let's take a look, and I'm going to give you full disclaimer. This analogy isn't mine. I learned it from one of my A&P students many years ago. Okay. So we're going to give social roles to the proteins here. In order to contract, actin filaments, thin filaments, and myosin filaments, which are thick, have to connect physically, right? So actin filaments that's the girlfriend. Myosin filaments, the boyfriend. They really want to hang out together. They can't. Because there's dad, tropomyosin, that blocks the active sites on actin and prevents myosin from interacting with it. But we all know who's in control of the dad. It's mom. It's troponin. So suddenly a family friend arrives, calcium. And calcium starts talking to mom. Okay? It binds to troponin. When it binds to troponin, when family friends start talking to mom, mom pulls dad into conversation. In other words, in response to calcium, tropomyosin is removed from the binding site, exposing them. Now, that isn't watching girlfriend anymore. Tropomyosin doesn't block actin. And the boyfriend and the girlfriend finally can hang out together. So the formation of so-called cross-bridge starts the cross-bridge cycle. This is the last step, excitation, contraction, coupling. Because now, any questions right now? I think it's now we need to figure out how does that, how does that work? The contraction, the filaments, thin filaments being pulled in the process known as cross-bridge cycle. Since it's a cycle, it doesn't have a beginning, so we're going to randomly assign the beginning step. Here, you can see myosin that is bound to actin. And this myosin also has ADP and inorganic phosphate bound to it. Remember, just a quick reminder, the reaction of ATP hydrolysis, folks. ATP, when you add water and enzymes, is broken down to ADP, inorganic phosphate, and energy. Let me write this so it looks better. So ATP is broken into ADP, inorganic phosphate, and that breakdown also produces energy. This energy has been used to pull thick filament, and the term is cock it. Just like you, now correct me if I'm wrong, I'm not that good with the gun anatomy. Like you cocking back a trigger or whatever it is on a gun, that's the same, like single action, same idea. It is charged as a spring, the myosin head, and when the myosin head releases ADP and phosphate, it snaps forward, sliding the thin filament relatively to thick filament towards the M line. This process, the sliding, is called a power stroke. Now we have a conundrum. Myosin heads propelled the actin towards the M line, but they still get attached, stay attached to the actin. We need to separate them. So how do we do it? This. Without ATP, myosin heads will not detach, which means we're going to get to rigor mortis, don't worry, which means we need to give them ATP. Once ATP binds to the myosin head separates from actin. The next step is the breakdown of ATP. Okay? Right here you can see that ATP hydrolysis, the breakdown of ATP, coax the myosin head back. Charging the myosin head so it is able to form a cross bridge again. Okay? That make sense? I'm going to move the to form the cross bridge again. And start the process of contraction from scratch. Okay? So, a couple of things. I mentioned when we were talking about thickened filaments, I mentioned the two main functions of myosin head is to bind ATP and break it down, and bind actin. In this process, you can see that both requirements are fulfilled. How long can muscle contraction occur? Your muscle contraction will occur as long as calcium and ATP are available. You may wonder why your muscles are not in a continuous state of contraction. Remember that action potential that we talked about that releases calcium? It's not endless. When that electrical charge through the membrane is over, calcium is pumped back into sarcoplasmic reticulum, and that's it. That's the end of the contraction. Getting there? No, that's for dead. That's for dead bodies, okay? So, here's the deal. When cells die, one of the things that cells do when they die, they take in calcium. At the same time, they don't produce ATP anymore. You know, they're dead. So on whatever's left, they form cross bridges. Okay? And that results in a weak contraction of your skeletal muscle, so-called rigor mortis, which peaks at about 12 hours post-mortem. I don't know if you watched, I'm going to try the newer show, Reacher, on Amazon. I have a very specific taste. Yeah? Okay. Anyway, so main character is a very strong guy, let's put it this way. And he kills a bunch of bad people. And in one episode, he kills 2 bad people, and then comes back later, like, to move the bodies. And bodies are in the rigor mortis. They got pretty accurate with that. And he basically folds them in the trunk, and you can hear the folding process. Yeah. Eventually, though, cross bridges are destroyed by the enzymes within the cell. Now, calcium levels are pretty important for the regulation of your muscle activities. What's interesting, hypocalcemia, you may have guessed, oh, not enough calcium, muscle is not going to contract, but turns out, no. Calcium regulates the transport of sodium, so extracellular calcium, when low, results in basically contractures and tetany. Hypercalcemia, on the contrary, makes muscles irresponsive. Now we're going to get to nitty-gritty of the tension that's produced during contraction. So there are two types of muscle contractions. Isometric and isotonic. When contraction in isotonic, it means that the tension, the force that muscle produces is greater than the load. When contraction is isometric, tension is lower than load, and it means muscle doesn't change the length. Three factors that determine the tension, the force of the contraction are frequency of the stimulus, load, size of the fiber. So before we figure out what determines the force of contraction, we need to introduce the concept of a muscle twitch. As we mentioned before, every motor neuron innervates several muscle fibers. Okay? Very much like we can think of this class as a motor unit. And I'm not trying to be like dismissive or funny. You are listening to me. In this situation, I am a motor neuron, and you are muscle fibers. As you can imagine, the class can be very small, or it can be very large. Now, which class is easier to manage? Smaller or larger? Smaller. We will get back to this idea that smaller motor units provide fine controls. There is some trade-off. So, the contraction of a single motor unit in response to a single action potential is called a muscle twitch. Muscle twitch has a latent period. That's excitation part, when action potential travels through the membrane. Contraction part and the relaxation part. Two things about motor units. First of all, within the same muscle, motor units contract asynchronously. When one contracts, another one rests. Another important feature is that motor units are different. Sorry, not motor units. Muscle twitches are different for different muscles. You can see here in this image that extrinsic eye muscles, such as lateral rectus, have an extremely fast contraction. It's, pretty logical that the muscle responsible for the eye movements contract faster. Gastrocnemius, a muscle that forms your calves, it's also pretty fast, jumping. And then this is actually a very good example, soleus. Soleus muscle is the deep muscle of your lower leg, the dorsal aspect of it. It actually is next to gastrocnemius. But the main function of soleus is posture. Posture doesn't require a quick contraction, and you can see that the twitch of soleus is pretty slow. So as we mentioned before, one of the mechanisms that regulates the force of contraction is the frequency of stimulus. This works, listen, exactly like the temporal summation on the synapse in the neurons. So when single twitch is produced in response to a single stimulus, the maximal tension is limited by the strength of that motor unit and the force, and it completely relaxes. When you start overlapping single twitches, effectively having a temporal summation, the force of contraction dramatically increases. You can see it here. Okay? And this situation, when, as you can see, there is a period of partial relaxation between individual twitches, we have on our hands unfused or incomplete tetanus at the maximum frequency of stimuli. There is absolutely no relaxation between individual twitches, which allows your muscle to get into the fused or complete tetanus muscle achieves, motor unit achieves its maximum force. But you can imagine that if this motor unit works sort of over time, not over, at its maximum capacity, it gets fatigued pretty quickly. Now, another parameter that can increase the force of contraction is the strength of the stimulus. In this case, we're going to switch from looking at a single unit at multiple. We're going to look at multiple units. So when the signal strength, the stimulus strength, increases, the number of motor units that is excited also increases. When more motor units are activated, that inevitably increases the strength, the force of contraction. Obviously, there can be a stimulus that is not strong enough to activate even a single motor unit. In this case, you have no contraction at all. But there is another important feature that we need to address here. The number of motor units in a muscle is limited. So, at some point, when motor unit reaches its maximal strength and effectively stimulates all motor units within the muscle, all fibers, your muscle reaches its maximal contraction. First, motor units that are activated are smaller ones. Then we go to larger and larger, and the largest motor units are activated last. So now let's step back and just think about your different muscles. So let's consider quadriceps muscles. Are they strong? They are. I mean, I'm not saying that you have the strongest quads in the world, but relatively to other muscles of your body, they're pretty strong because they have, by and large, bigger motor units. Well, they're also bigger, but larger motor units. The effort that you can produce using the muscles controlling your fingers, flexor digitorum profundus or something like that. Are they strong, those muscles that move your fingers, compared to quads? No. Can you crochet, hypothetically, with your fingers? Can you crochet holding threads between your toes using only your quadriceps? I seriously doubt that. So there is a trade-off. Smaller motor units enable your muscles to be precise, to produce fine movements. But the trade-off is reduced strength. Larger motor units deliver strength, but have very limited precision in the movements that they can execute. Okay? Finally, which is super obvious, bigger units will produce stronger contractions. Because if you have larger fibers, I mean just simply bigger cells, it means that they have more myofibrils, which means they can generate more force. Okay? So the enlargement of a muscle, and I'm going to use gym terminology and gym experience all the time. If you look at people who, like bodybuilders, they strong. They are. Because they have huge muscles. Okay? So this graph on the bottom, for example, shows you that muscle hypertrophy increases the force, and that increased cross-sectional area is associated with a greater force. Muscles become bigger by the process called hypertrophy. I'm going to highlight that term. Now, hypertrophy refers to the fact that each cell becomes bigger, but the total number of cells remains the same. Your skeletal muscle cells do not divide. There are extremely rare instances of so-called hyperplasia in which new cells are formed. Again, we can discount that mechanism. So basically, when you look at a bodybuilder, you can feel better knowing that the number of muscle cells that they have and that you have is pretty much the same. Okay? Finally, the stretch of a muscle. Skeletal muscle cells have the optimal length for contraction. So if you look at the sarcomere at the resting length, thick and thin filaments have a pretty decent overlap. When you look at the sarcomeres that are stretched too much, the overlap isn't that good, so it's really hard to initiate a forceful contraction. On the opposite side, when the muscle is fully contracted, there is no more space to move anywhere, so it's really hard to produce forceful contraction as well. You can see it in a simple exercise, which is a pull-up. So imagine that you are hanging off of the pull-up bar. The muscles that will help you to pull yourself up, for the most part, the latissimus dorsi, rotator cuffs, a bunch of muscles of the back, traps, as well as the synergist of the biceps, they stretched. So the hardest part in doing a pull-up is to start it. If you start at not stretched, like somehow you manage to get yourself a bit of an up, it's much easier. And then when you get to the muscle maximum contraction, this is why doing a muscle up is so hard. Because your arms and your back cannot produce significant effort once you reach the top of the pull-up And this is why muscle up is not as much of a force exercise. It's a skill exercise. There's a bunch of people who definitely can do it from the strength side of things, but it requires a certain movement, certain flow to be able to do that. Okay? So, as we discussed, isometric versus isotonic. I'm not going to spend too much time here. What I want to highlight is this. Again, we're going to do a lot of gym references. First of all, isometric contraction. Force is lower than the load, so there's no movement. Are they useful from the training standpoint? Very much. They train strength. What I sometimes do, honestly, in the car, when I'm bored, I press my head against the headrest, to train the neck muscles. You can do exercises in a car. Just find some immovable part, not the handbrake, and you can work on your arms or whatever. Okay? Now, isotonic contractions, in which force is greater than the load, are divided into two subtypes, concentric and eccentric. In concentric contraction, muscle shortens. In eccentric contraction, muscle lengthens. So again, I don't know what your gym goals are, and if you have any, which is totally fine. But if you do, if your goal is pure strength, focus on concentric contractions. Some people who train strength, like seriously train strength, for instance, the easiest example is deadlift. They would do a deadlift and then drop the bar once they reach the top point. Am I making sense? They do not go down. Okay? Now, eccentric contractions are useful if you're working on the size, on the hypertrophy. So, for example, if you look at any hypertrophic training program, it focuses on the relatively short concentric and then long eccentric. Does that make sense? Now, this is one of the duh moments. So, I'm going to give you, hypothetically, a plastic bucket, small plastic bucket, like kid's bucket, that's filled with water, and a Home Depot-sized bucket that is filled with sand. Which one you're going to throw farther? Kid's water. Which one is going to fly faster? Kid's water. Because the contraction, the concentric contraction that you will be able to produce, depends on the load. The greater the load, the shorter the move, and the slower the move. Does that make sense?